To question 2. State the difference between excretion and elimination. And another word that we can use for elimination is egestion. So excretion is a removal of metabolic waste from the body, while elimination, using the word that they have, is the removal of undigested food material or undigested waste material from the body. So this is what comes from the alimentary canal. Anything that is not um, anything that is not absorbed is removed. So then we have part 2. Other than the kidney, name the 2 excretory organs in the body and state 1 excretory product from each named organ. So you have the lungs secretes, or not secretes, sorry, but gets rid of carbon dioxide. You also get rid of heat. This could be another option. Then you have the skin. It gets rid of urea. It gets rid of excess salts, excess water, and we know that these 3 combine to make sweat. Then we go on to part B. So here we have figure 3, which shows a diagram of a nephron of a kidney. So if you could recall, the nephrons is what is responsible for the actual filtration or the filtering of the blood. And in each kidney, we have millions of these nephrons. So you have this structure here, and what they require you to do, they have blood is filtered from the glomerulus, and the filtrate enters the Bowman's capsule. So list in the correct order the 4 structures after the Bowman's capsule through which the filtrate will pass as urine is being formed. So let's come back up to it. So if you look at this, we have the Bowman's capsule, you have the glomerulus. After the Bowman's capsule, you have this part here before you get down into this loop, and this is your proximal convoluted tubule. After the proximal convoluted tubule, you have the loop of Henle. After the loop of Henle, you have the distal convoluted tubule. And last but not least, you have the collecting duct. Remember that the collecting duct is what passes the now called urine into the ureter for it to get to the bladder. And then from the bladder, you get rid of it from your body through the urethra. Then on to the last part. So you have part C. So table 1 shows the concentration of 3 substances found in the filtrate and urine of a healthy individual. So you have substance, urea, glucose, protein, the concentration in grams per cm cube, you have filtrate in the Bowman's capsule, and then you have the urine. So you have 0.5 urea in the filtrate, while you have 15 in the urine. Glucose 4.0 in the filtrate, but you have 0 in the urine. And then proteins, you have 0 in both the filtrate and the urine. So part 1, explain why glucose is found in the filtrate but not in the urine. So if you could recall, there are 2 parts to this whole filtration process. You have ultrafiltration, and then you have the reabsorption phase. So with ultrafiltration, this is where the pressure coming from the blood that is transported or coming in via the renal artery, when it gets to the glomerulus is made up of a network of capillaries. So the capillaries being the smallest blood vessels, but the pressure is still high because, if you could recall, the renal artery gets its supply directly from the main artery coming from the heart, which is the aorta. So blood is coming under very high pressure. The pressure is not changing, but the size of the blood vessel is changing. So when we go from artery in terms of size. So we're going from artery to arteriole to capillaries. The size is getting significantly smaller, but the pressure is remaining the same. So at the glomerulus, the pressure is high enough to forcefully squeeze out any substances or any particles from the blood that is small enough to pass through the walls of the capillary. So when this happens, this is where we have things like, for example, the main things that we mention is urea. You have, of course, the water. You have salts, which is the ions. You have glucose as well, and hormones. So glucose is small enough to be squeezed through the glomerulus through that process of ultrafiltration. So this is why we will find glucose in the filtrate. But then the urine, which is completely formed when it gets to the collecting duct, you don't have any glucose there. But way before the collecting duct, you are not supposed to have any glucose, and that is because the glucose is selectively reabsorbed through the process of using active transport at the proximal convoluted tubule. So right after the Bowman's capsule, if you could just get back to this diagram. So right after the Bowman's capsule, where the filtrate enters, that first part after the Bowman's capsule, which is your proximal convoluted tubule, this is where all of the glucose in a healthy person with functional kidneys is supposed to be selectively reabsorbed into the blood. So going forward to the loop of Henle, you shouldn't have any glucose, and therefore you definitely should not have any glucose in the urine. So this is what accounts for that. And the last part, explain why protein is found in neither the filtrate nor the urine. Getting back to the fact that we said in ultrafiltration, it is the particles that are small enough to squeeze through the walls of the capillaries. That is what will get into the Bowman's capsule to form the filtrate. Proteins are too large. So the proteins are too large to squeeze through the glomerulus. Thus, this is why we don't find proteins in either the filtrate or the urine.